For my research, I've sort of focused on fetal and newborn development and the brain. And for environment, I've gone from chemical pollutants, which was my initial love, to actually endogenous neurotoxicants that every baby is exposed to, like bilirubin. And I look at it from a range of points of study, one of which would be epidemiology on one end, where I'm looking at large groups of people and exposures to see what the outcome of pregnancy is, all the way to the other extreme, which is to work in my laboratory with cells and molecules and try to understand the basic mechanisms of how those exposures are affecting brain development so we can intervene and improve outcomes of our babies. We know that fetal alcohol exposure is the leading cause of developmental disabilities in this country and around the world. So I wanted to intervene on that problem also. If we intervened on moms who we identified as drinking during pregnancy, if we gave them just a telephone interview, if we gave them more intensive counseling, or if we did inpatient treatment, we found that the development of this test is like any newborn screening test, that the cost of it is small compared to the healthcare savings that you have once you make the discovery that a baby's been exposed to alcohol. We haven't just looked at alcohol, we've actually looked at other air pollutant type solvents. So we study these solvents because we think the mechanism is through a solvent action. You know, like if you get fat on your clothes, like you spill grease or something, you get it out with a solvent because it dissolves the grease. Well, all your cells are covered with a grease layer called your plasma membrane, and parts of it are very important for how the cell functions. And so we think that solvents are acting on those grease bags to inhibit their action. And so we think that choline has some effect on the fats that are in the cell membrane that these solvents are affecting. So we've looked at toluene, which is an important solvent in terms of household cleaners. We found how levels of its metabolite in the urine of our babies in the NICU who are in isolettes. And we find that it goes through the same type of mechanism that alcohol goes through. We've also been looking at bilirubin, which is, you know, your body makes bilirubin, you're not like exposed to it, but our preterm babies have trouble dealing with the bilirubin. And we're beginning to recognize that at much lower levels of bilirubin than in a term newborn baby is affecting the brain function of those babies. And so, again, in my animal models, I can show that if I supplement them with this choline, that I improve their ability to do certain tasks or their behavior. So I think one of the end games is looking at, one, how choline is exerting this, and two, starting to look at whether choline supplementation in the NICU is improving the outcomes of our babies in the NICU. And maybe the third one would be if we could give choline to all pregnant women, make sure they're getting enough, just like folic acid, could we actually improve the outcomes of women who are exposed to alcohol either knowingly or we're all exposed to alcohol unknowingly because it's just part of our environment. The long-term goal is to improve the outcomes of pregnancy, particularly in terms of the cognitive and mental abilities of the babies that are the result of those pregnancies.